Thank you, Maya. Let me start sharing my screen here. Can you see my slides?
Yes, perfect.
Yeah. Okay. Yeah. Thank you, Elucidata team, for the invitation to speak in this forum. And I'm Diwakar Sangaraju, and I'm a senior scientist at Genentech. Our team is focused in application of metabolomics to various drug research, discovery, and development projects. And today I'm going to talk about this high throughput metabolomics data analysis using Poly platform to uncover this metabolite signatures to better understand biological processes. So we are definitely in an omics era where we can probe and quantify each and every aspect of the central dogma, ranging from gene to protein expression, using various omics technology platforms. And all of this data, we are using it to explain the final phenotype or the phenome. For example, basically comparing disease versus healthy or drug treatment versus control. And metabolites, they sit in this very special position above the phenome or the phenotype, where the ligands or the metabolites can interact with these enzymes or proteins to fundamentally elucidate a phenotype. And to define metabolomics, I mean, it refers to a systematic identification as well as quantitation of these small molecule metabolic products. Together, we call the metabolome or lipidome, which is a subset of metabolome, of this biological system at a specific point in time whenever we sample that biological system. To simply put it, this entire data from genomics to proteomics can explain what might happen in a specific biological system, but metabolite information along with other omics data can better explain what is happening currently in that biological system in that particular point in time. So all of this omics platforms, they are possible, again, because of the rapid advancement in technology and instrumentation, which can enable us to generate a lot more data points nowadays. So it is well and good, right? Metabolite data is very important for us, so why not go ahead and analyze them? Well, we quickly realized that there are some couple of challenges here. There is an analytical challenge and complexity due to the diverse chemical structures of these metabolites. And not only that, there is also this challenge of faster data analysis to reach ultimate biological insights. So what do I mean by this analytical complexity, right? If you think about proteins or genes or RNA, they are essentially polymers or repetitions of some basic monomeric units, where the structures are pretty defined and their analysis is relatively less complex as compared to human metabolome, which is very diverse. I mean, like there are structures that range from very low molecular weights below 100 to way over 1,000. And there are more than 100K diverse compounds, which kind of present this analytical challenge with respect to metabolite quantitation and analysis. So what is the scope of metabolomics in pharma or, for example, in biopharma R&D? So if you think about it, mostly we are interested in knowing the changes or perturbations in the known metabolome or a lipidome in a biological system. Essentially, what is happening to this metabolic skeleton in a specific context, like, for example, drug-treated versus control, or one animal model versus another animal model. So we are interested in knowing this known metabolites because the unknown metabolite space is a very complex problem to solve, and it takes a lot of time to essentially evaluate it. But we feel that there are many more interesting features that we can learn from this known metabolome in this various contexts. And these include small molecule metabolites ranging from like 400 to 500 metabolites that define the basic metabolic skeleton which have a molecular weight of around less than 600, or even this complex lipid species, which are around in thousands of them, that can be characterized by this standardized lipidomics platforms. And for specific projects like which needs specific groups of metabolite characterization or their analysis in a quantitative manner, we have several targeted quantitative metabolic panels that can be applied to those projects. So using these basic tools that are available to us, which can verify and probe the known metabolome, we want to understand whether there is a metabolic data signature that can rapidly teach us something about certain aspects of target biology or drug toxicity or in vitro cell culture, for example. So some of these things include, like, for example, if you have groups that are control versus drug treatment, we wanted to know what are the metabolic signature patterns that are changing with respect to acute drug toxicity, or what is the pattern change with respect to polypharmacology of a certain investigational drug. Like, for example, what is target 1 versus target 2 changes in pharmacology. Not only that, metabolomics is increasingly being applied to bioprocess characterization, especially in the context of a therapeutic antibody production process, which is a biological process using mammalian cell cultures. So we want to characterize metabolomics more in that area, or the changes in metabolite signatures. And also at the research or the basic research level, we want to define biology or compare animal models, like animal model 1 versus 2, which are not well characterized in certain therapeutic areas, such as neurological diseases, right? And not only that, even applying it to in vitro target evaluation. For example, if you have an expression system that is overexpressing a certain target, we want to understand metabolomics in that system as compared to the control system. So to fastly analyze this data and predict this data signature patterns, we wanted to quickly scan for all of these metabolites, the known metabolome, and select for this top 100 to 200 metabolites that dictate these changes. For example, in this heat map, we are comparing between like a control versus a compound 1, investigational compound 1 versus investigational compound 2 within the same subclass of molecules that are used for the treatment against a target 1. So this can distinguish these compounds with respect to this metabolite signatures. And search for these metabolite signatures probably as an early indicator of a later phenotype. It can be a tox signal or it can be activity, but we wanted to understand these signatures robustly at this level. And also see if this metabolite signature can provide any additional evidence along with other omics data, like that is typically collected in some of the in vitro experiments, such as RNA-seq data. And we really wanted to do this at this level in a very faster turnaround time within 2 to 3 weeks after we receive a request from the project. So, or even like think about this real-time in vitro cell culture monitoring maybe in the future. So the reason behind this is teams are very agile and they're very nimble with respect to getting insights pretty quickly on whatever data that they're collecting. And we want to enable those teams to make such decisions in a very faster turnaround manner. Because if it is a longer time, then they would have moved to another question, and the problem becomes insignificant pretty quickly. And this top 100 to 200 metabolite changes with changes which are changing with respect to a certain question that we are asking or that are not changing, we wanted to evaluate that signature much more robustly. Like, is there a reproducibility and confidence in that signature? Before we go into predicting this or understanding the biological pathway changes that are happening with respect to that signature. So first, we wanted to reproduce the signature over and over again, like for example, in that particular series of compounds or the class of compounds that we are testing. Ultimately, the signature should definitely be correlated to a specific phenotype, which can happen later in that biological system. So the initial focus we wanted to spend on the robustness of the signatures of these known metabolites in various contexts, and build probably a predictive pattern recognition and modeling to better define compounds from the metabolomics point of view, apart from the other data points in other omics that we are collecting generally. Only then we want to move into this biological insights, right? Because biological insights with respect to pathway changes is a very complex problem to solve, and it requires a lot of time. And it's also experiment dependent, whether we are investigating in vitro or in vivo, where the changes might be happening globally and it might not reflect a particular tissue or a specific region of change, right? And having this biological insight, I would say, is definitely beneficial. It's great to have, but metabolomics alone cannot define this. There has to be this connection of metabolomics, which is a sparse data set, to a rich data set such as transcriptomics data. And this is definitely a collaborative team effort at this stage, once we know that the signature changes are robust. And because this is time consuming, we really wanted to push this to the very end once we are confident about the signature by repetitive experimentation. And within, for the analysis side of things, there are pretty much standardized methods that are available now for metabolomics evaluation, at least for known metabolome analysis. It starts from study design all the way to scientific reporting. So in our case, we deal with a lot of different type of samples within our team, and we have defined sample preparation techniques that can enable multi-omics analysis within the same sample, such as metabolomics, proteomics, and lipidomics within the same sample, and also enable different workflows. Either it can be a global untargeted workflow or a global targeted workflow, or a specific pathway targeted, or even like a couple of biomarker validated assays. And all of this, we are enabling it through complementary HPLC analysis techniques, including helic and reverse phase chromatography, to evaluate a wider or a broader characterization of this known metabolome. And the final step is, and a very important step, I would say, is the data processing and scientific reporting, which kind of takes up a lot of time. We really wanted to accelerate this process to reach our goal there with respect to reporting data to the teams within this 2 to 3 week time frame. And for LCMS data processing, we use a variety of tools that are vendor tools that are available, and they are improvements made with respect to these tools from one generation to another generation, which can enable this high throughput LCMS data processing. So after data processing, you end up with this big data sets of LCMS data to get insights that are reportable, right? And for this, we kind of partnered with Elucidata, especially using their Poly platform that enabled this through a series of apps that are deployed in Poly pretty quickly. That was almost like a plug and play for us to enable this type of data insights and reporting. And we also use some sort of coding to get to those customized versions of those reports. So Elucidata Poly platform, like for example, if you think about it, it's essentially an end-to-end platform that was there. The mass spec data processing of it, I think there is an option for the user to either use LMaven or the vendor tools. I think we are using vendor tools because we are just familiar with the tools, and we are trained in those tools for a certain amount of time. The users are trained there. And after we get this LCMS data processing, we put it through the Poly to various tools that are available in Poly to quickly get to this characterization or quality check for the batches, such as sample pool QC and internal standard quality checks, and quickly plot the data as a differential expression using variety of plots using tools available in Poly. So one tool that I want to highlight here is the Poly dual mode data visualization tool. That is a Metabolomics app tool that can give us access to different types of visualization in one go after we get to those peak intensity tables from the vendor tools. So these include either the PCA plots, two-dimensional or three-dimensional plots, just to see how much of a variability there is between the groups and how differentiating they are in that experiment. Not only that, even volcano plots, seeing the significant changes in one snapshot of the metabolites that are changing, and even to the micro level of the different trends of metabolite changes with respect to a bar plot with stats. And not only that, I think the heat maps that they have deployed are also very useful within the same tool, and we are able to communicate all of this, as I will show, in a reportable format to the teams. Like, for example, we very much like this offline HTML-based reporting, where, sorry, I think the video is not playing here. Let me play the video here. Okay, yeah, where we can plug all the data and the insights that we are seeing with respect to the study in a very organized way, so that teams can have access to this complete data with the visualization in an offline HTML report that is easily navigatable, which kind of puts us in a platform for a further discussion and planning additional experiments or evaluating additional hypotheses based on this initial data set. And again, like, the Poly platform is also a project management platform. So we can get projects managed within the Poly platform and have data imported into it for further evaluation using various tools that are available there within Poly. And not only that, like, we also wanted to take advantage of some of the publicly available metabolomics data that is well curated in some in databases such as Metabolites or Metabolomics Workbench. And we were able, I think, Elucidata was able to get this curated data within Metabolomics Omics Atlas, which has extensive number of data sets that are well curated with meaningful insights that are pushed through the Data Studio app and getting to those customized visualizations that I showed before. And it's again like navigatable data, which is organized according to the disease type or the tissue and whatnot with respect to subcategorization of this data. And this is very useful for us to get probably answering very basic questions that teams poses, like what are these important metabolites that come up in a certain disease type without even performing the actual experiment. And apart from those above two tools, like we also partnered with them to get some customized data analysis tools that we see are interesting based on the literature that is published recently. For example, if you come across literature and there is some tool that is interesting for us, we quickly are able to reach out to Poly. Like, for example, in this case, this is a tool that is published by Dr. Paris' lab, which is evaluation of ToxEMS, which kind of understands the changes of metabolite levels in a dose-dependent manner in an in vitro system. So this type of evaluation can enable dose response metabolomics, for example, not only metabolites that are related directly to the activity of a drug, but also metabolites that are perturbed because of the polypharmacology of a drug. And essentially, this tool that they published is a pattern recognition tool with respect to changes in EC50 of the drug, either an increase or decrease in EC50s, or a random pattern. So they published this code as part of the publication, and we quickly reached out to the Elucidata team to see if we can get a customized tool deployed for us, because we are not well versed with respect to executing code. They were able to execute the code and deploy this tool in a simple UI so that we can use that for internal evaluation of a specific exploratory proof of concept projects. So with that, there are definitely various tools that we got access to within the Poly platform. They range from the ToxCMS tool that I just spoke about to the Metabolomics data analysis tool, or we have a different tool for lipidomics data analysis, or dual mode data visualization tool, and also this data lakes as part of this Metabolomics Omics Atlas. So ultimately, like as we build a lot of projects within the Poly platform using the project management, our goal is to ultimately come up with internal metabolomics omics atlas that is specific for Genentech projects that are managed within Poly. So with that, I think I would like to acknowledge the Genentech team, who both the metabolomics and lipidomics teams at Genentech, as well as the Elucidata team for supporting us and to enable this slew of tools that can make the data analysis for metabolomics faster.